In this video, we're going to talk about how genetics is more complicated than what Mendel had proposed. Now, the law of segregation and the law of independent assortment, they still apply. There's nothing that goes against that. There's still a direct result from what happens during meiosis in spermatogenesis and oogenesis. So together that's called gametogenesis. So nothing takes away from that. So what we're going to do is we're going to show how what Mendelian genetics is, and then how it could be a little bit different. So, for example, that in Mendelian genetics, when you were looking at two alleles of a gene, he always found this dominant recessive relationship, that one of the alleles was dominant to the other. But what we've learned is that's not always the case. There are other relationships that can occur between two alleles of the same gene. The first is complete dominance, and that means in a heterozygous state. So if you've got an individual that has both of the two alleles of a particular gene, that only the phenotype from one allele is seen. So this is your dominant recessive relationship that we've already discussed. So how do we figure out which of the alleles is dominant and which of the alleles is recessive? What we do is we have a pure breeding strain here of one of the alleles, and so in this case the big R allele, and that gives rise to the trait of round seeds. And we've got a pure breeding strain here. No matter how many times you take the offspring and cross them to themselves, you're always going to get round seeds. And we do the same for the other allele. Okay, we've got a pure breeding little r allele, or offspring, organism. And when you cross them to each other, you get of course, a wrinkled offspring. And if you breed the offspring together, you're going to constantly get the wrinkled seeds. So they are pure breeding. So you take pure breeding for one of the traits, you take a pure breeding strain from the other traits, you cross the two together so that you know 100% of the offspring are going to be heterozygous. You look at the phenotype there, round. It is identical to one of the two parental phenotypes, this one here, and therefore the round trait is dominant to the wrinkled trait. The round allele is dominant to the wrinkled allele. Okay, so this is the sort of experiment that Mendel did to define which trait was dominant and which trait was recessive. All right, now let's look at how things can be a little bit more complicated. In some genes, when you take those two alleles and when you put the two different alleles into the same organism, so in that heterozygous state, the phenotype you get doesn't look like either of the two parents. Doesn't look like that from either of the two alleles. So let's take a look. Here we have flowers. And we have pure breeding red flowers so that we know that they are homozygous for this red color. We have pure breeding white flowers, so we know that they are homozygous for this white allele. And when you cross the homozygous red to the homozygous white, you get 100% of the offspring, which we know are heterozygous. They must be heterozygous, are neither red nor white. They're pink. They're a completely different phenotype. And so we say that the CR allele and the CW allele of the flower color gene are incompletely dominant. Because when you have a heterozygous state, you see a new phenotype. You see a third phenotype that is not in the mom, that is not in the dad. Now, that doesn't mean that we are blending these two together. And if you were to take two pink flowers, right, that are heterozygous and cross them to each other, you're going to see white coming out 1/4 of the time. You're going to see red flowers coming back out 1/4 of the time. So you can tell if you take a gene that has two alleles that have this incomplete dominance relationship, and you cross them in a monohybrid cross, you're going to see this phenotypic ratio, 1 red, so 1 to 2 to 1. Okay, that's different than the 3 to 1 that you see in a monohybrid cross from two alleles that share a dominant recessive relationship. All right, let's continue. Well, let's explain this first in molecular biology terms here. What are the let's get to the proteins that these alleles encode. And the idea here is this, is that the CR allele encodes a functional enzyme that generates red pigment. And so, if you are CR, you make enough of this enzyme to convert enough of a colorless intermediate molecule into a red pigment, and so you get a red flower. Now, the CW allele encodes a protein in enzyme that does not function. It cannot catalyze the production of red pigment. So if all you have is that broken enzyme, so 2 copies of that broken enzyme, you end up with no red pigment at all and therefore get a white flower. Now, if you are in a heterozygous state, you are going to have 1 dose of the properly functioning enzyme, whoops, over here, the CR. You're going to have 1 dose of the broken enzyme, and so you have half the amount of working enzyme that you do in a situation like this when you're homozygous. So that makes just half the amount of red pigment, which means you end up looking like this. You have a pink flower. So this is the sort of the molecular and the biochemical reason for incomplete dominance in this particular example. Now, the third and final possible relationship between 2 alleles is codominance, which is in a heterozygous state, the phenotypes of both alleles are present. And this is best viewed in blood types of humans. So there is an enzyme called I1 branching 1,6 and this. Big, giant, long word, but it's an enzyme. And it's an enzyme that catalyzes the production of carbohydrates so these glycoproteins on the surface of red blood cells. Now, the IA allele and the IB allele both encode functional enzymes. But the difference is the IA allele encodes an enzyme that makes a very specific glycoprotein. We're going to abstractly show that as a triangle. An IB enzyme encodes an enzyme that does a slightly different glycoprotein that we'll show with a circle here. So if you take 2 individuals, one who is homozygous for IB, one is homozygous for IA, you cross them to one another and you look at the offspring who is going to be, 100% of the offspring is going to be heterozygous. And you can see on the surface of that red blood cell, you can see, right, the phenotype from one parent, you can see the phenotype from the other parent. This is now codominant. You see phenotypes from both parents in the offspring. And that defines a codominant relationship between the IA allele and the IB allele. Okay, next, Mendelian genetics, each character was controlled by a gene with only 2 alleles. So a character like flower color was controlled by 2 flower color alleles, the purple allele and the white allele. Well, it turns out that most genes actually have way more than just 2 alleles. And one of the best examples of that is we're going to come right back to the human eye gene. The human eye gene that controls blood type actually has 3 different alleles. It has the IA and the IB like we've already seen, but it also has the little I. Now the little I encodes an enzyme that doesn't work at all, so you don't get any glycoproteins on the surface of the red blood cell. And what that means then is the I the little I is recessive when you compare it to both the IB and the IA. Because in this recessive, heterozygous state, IB, you get red blood cells with the B glycoprotein. In a heterozygous state like little IA, you get a red blood cell with the A glycoproteins. So when you have a gene that has multiple alleles, okay, you have to look at the relationship between each one in various pairwise comparisons. Another Mendelian trait is a Mendelian genetics here, is that one gene affects one character. So the flower color gene affects flower color. The seed-shaped gene affects the shape of seeds. But that's not always the case. Sometimes there are genes that affect multiple characters, and that is referred to as pleiotropy. So if we take a look at the hemoglobin gene, the hemoglobin gene controls really of all of these things, because what happens here is if you have the normal allele of the hemoglobin gene, the hemoglobin proteins will be normal, the red blood cells will have a normal shape, and you won't have any problems with all of this down here. However, if you inherit the sickle cell allele, if you're homozygous for the sickle cell allele, that's going to cause the hemoglobin's molecules to clump together and form fibers. That is, distend and cause the red blood cells to form the sickle cell. They will then lyse more easily, so you'll have anemia. They'll get clogged and clump in the blood vessels, and so you'll have these fever and pain and damage to various tissues. And so all of these various traits occur from one gene. That's called pleiotropy, one gene controlling more than one character. Now, the reverse can also happen. So in Mendelian genetics, one character is controlled by one gene, right? So your flower color is only controlled by the flower color gene. The seed shape is only controlled by the seed shape gene. Well, that's not always the case. In fact, there are lots of character out there that are controlled by many different genes, and that's called polygenic inheritance. And the most common form of type of character that will be polygenic are the quantitative characters. The characters that don't exist as just as a black and a white, but everything in between. So if we look at all of these runners here, we see their height, right? We don't just have two separate heights. We have everything in between. We don't have just two separate skin colors. We have all the gradations in between. These are called quantitative characters, and these are the type of characters that are most often controlled by more than one gene. Here would be a simplified way in which skin color might be represented, where we have 1, 2, 3 different genes that control skin color: the A gene, the B gene, and the C gene. The the big allele of each one of those gives rise to darker skin color, and the smaller allele gives rise to the lighter skin color. So that you have these two extremes and then everything in between, the two extremes being dominant for all three. So big A, big B, big B, big C, big C. That's the darkest skin. And then you've got the other extreme here where you're recessive for all three: little a, little b, little c, and you are as light as you're going to get. And then you have everything in between giving us a gradation, so we don't just have two different traits in this particular character. We have a gradation of traits. Now, another thing that we didn't see with Mendelian genetics is that in Mendelian genetics, the phenotype that you see from one gene does not affect the phenotypes from another. And that's not always the case. There are examples where the phenotype from some genes can mask the phenotype from another gene, where because of the phenotype from one gene, you can't tell what the phenotype is of the other gene. That's called epistasis. And oftentimes, epistasis occurs when the two separate genes that we're looking at are involved in the same biochemical pathway. So for example, let's take a look at this biochemical pathway that gives rise to a black pigment molecule. And so what you need is you need enzyme C here that will turn this colorless pigment molecule into a brown intermediate. And then you need enzyme B that will convert this brown intermediate into a black pigment molecule. So if let's say that each of these two genes are proteins are encoded by two separate genes, so we've got the color gene encodes enzyme C, and we have the black gene that encodes enzyme B. Okay? But right, you need that color gene to make enzyme C to get that brown pigment. Without it, there's nothing for enzyme B to turn into a black pigment. And this is the basis of epistasis. The basis of epistasis. That's pretty good. All right, so let's take a look. Let's say here that we've got, so there's our B, which gives rise to, right, the enzyme necessary to make the black pigment. And it is dominant. There's two versions of it. There's one version that makes functional enzyme B. That's the big B. And there's a version of it that makes non-functional enzyme B. That's little b. Same thing here with enzyme C. There's a color gene that gives rise to a functional enzyme C. That's going to be the big C. And then there is an allele of this gene that gives rise to a non-functional enzyme C. We call that allele little c. So when we do a dihybrid cross, so two black mice to each other that are dihybrid, meaning they are heterozygous at the B locus and heterozygous at the C locus, and we cross them together, normally you would expect to see this 9:3 to 3:1 ratio, but here you don't, because the little c allele is epistatic to the B gene. Then even though based on the B genotype in these three mice, you would expect to see black mice, but because the little c is epistatic, instead you're seeing that trait, the white mouse. So these three would normally be black mice. Well, instead they are white mice. So now you get a strange, this new phenotypic ratio of 9 black mice to 3 brown mice to 4 white mice. Now, lastly, in Mendelian genetics, a given genotype will always produce the same phenotype. So if you were homozygous for the purple flower-colored allele, you would 100% of the time have purple flowers. But sometimes that's not the case. Sometimes there is an environmental influence on the phenotypes, that even though you have that same genotype in various situations, the same genotype in various situations, the phenotypes might be different based on environmental interactions. One of the best examples of that is hydrangea. Depending upon whether you grow up those flowers in basic conditions, in basic soil, or acidic soil, you can get two different flower colors, even though these two plants are 100% genetically identical. So we have the example here where we have the same genotype, okay, in the DNA of these flowers that are giving rise to two different phenotypes because of the environment in which they have developed. Now, that's referred to as the norm of reaction. And some phenotypes have a very limited norm of reaction. That means that there is really no influence of the environment on the outcome. So for example, if you are IA, you're going to have the A blood type regardless of the environment. But something like hydrangea here has a more broad norm of reaction, meaning that your trait, okay, is going to sometimes be different, even though you have the same genotype, might be different depending upon the environment. So skin color will have a broad norm of reaction, right? If you grew up out on the deserts in, let's say, Australia, you're going to have darker skin than if you grew up in some cloudy environment, maybe Scotland, something like that. So that would have a sort of broad norm of reaction, a high influence of nature on or environment on the phenotype. Okay, so the next lecture and the last lecture of this course is going to discuss the idea of genes being linked, so no longer assorting independently, and the idea of sex linkage, genes that are on the same chromosomes that determine the sex of the offspring.